All right, great. Thank you for the introduction. I'm Jaewan. I'm a postdoc at the Jose Avalos Lab at Chemical, Biological, and Engineering. And today I want to talk to you about shedding light on microbes. So using optogenetics to apply for metabolic engineering applications. So first of all, what's metabolic engineering? Do you have a laser pointer?
Oh, I do not have a laser.
All right, that's fine. Okay. So
Actually, yeah, if you use that, then people on Zoom can see it.
yeah, okay. So metabolic engineering is basically production of valuable chemicals from renewable substrates, such as sucrose, starch, or cellulosic biomass. And using that, you can produce things like bulk chemicals, therapeutics, biofuels, and important natural products. And how you do that is rewiring microbial metabolism. You can either rewire or you can put in a synthetic synthetic pathway into different microorganisms, such as bacteria or yeast, to produce as much valuable chemical as possible. Now, traditionally, biosynthetic pathway is introduced into a single strain of bacteria or yeast. Now, what happens is when you do that, you tend to strain bacteria or strain microbes with that one biosynthetic pathway that results in defects in growth. And as a result, productivity decreases. The idea is having co-culture. So in microbial co-culture or consortia, you divide that biosynthetic pathway into several components, into different metabolic modules, and put them into different strains of microbes. Now, when you do that, you divide labor amongst different strains of microbes, which allows you to lower metabolic placed on each strain. And also by dividing metabolic module into different strain, you can optimize each module in that specific strain, which harnesses the power of that particular strain in making that intermediate. And lastly, because you're dividing these modules, you can actually avoid endogenous regulatory mechanisms that exist when you put everything in one strain. Meaning maybe an intermediate, let's say in that green rectangle there, can actually inhibit the production of that triangular molecule due to endogenous regulatory mechanisms. Now, what's the problem with having microbial co-culture? It's that when you have microbial co-culture with different strains, one strain tends to take over the culture. It ends up outgrowing the entire process, meaning population stability is hard to achieve. Now, you can make different strains codependent on each other by sharing resources or codependent on each other for survival. The problem is alteration during mid-fermentation. So trying to change population ratio during fermentation is impossible when you do that. And whatever you have that you get, the population ratio you get from making them being codependent on each other is what you get. That's not the optimal population ratio to produce, to have maximal yield for that particular chemical. And what's the solution to that is optogenetics. So just to go over what optogenetics is a manipulation of biological processes using light-sensitive proteins. You can go from controlling transcription with light. You can also control neuronal signaling with light. What we want to focus on is light-inducible transcription. Here, that previous PhD graduate has developed from the lab, OptoEXP. It's a dark-inducible transcription factor that doesn't bind its cognate DNA in dark. Now, only when you shine blue light, that thing comes and binds its cognate promoter, making any gene of interest. Now, there's also a way to invert this signal where you have dark inducible transcription. Here you utilize the repressor activator circuit that's commonly used in yeast. GAL4, which is transcriptional activator, GAL80, which is a transcriptional repressor of GAL4. So you make GAL80 with light, and once you shine light, GAL80 is made, GAL80 binds to GAL4, no GFP expression. Now you can imagine GFP being other things that's relevant. So how do we apply optogenetics in metabolic engineering? So basic idea is this. So you have pyruvate decarboxylase, PDC1, which is an essential gene in baker's yeast. That you can control its expression with light. So in the dark, PDC1 is not made, meaning cells won't grow on glucose. Now, only when you shine blue light, cells will start growing. So with that, you're able to actually control different strains of yeast, their growth rate, with just dosing different lights, different amounts of light. And again, so as a result, the population dynamic, so population ratio in a co-culture, having two different strains, you can actually control the growth rate of each of them just by having different cycles of light, different intensities of light. And as I mentioned, this could also be dark inducible with the circuit I explained to you in the previous slide. But the problem is you can't go any more than having two strains, one grow with dark, one grow with light. You can't have more than that. So the idea is to have, what do? We add more light to this. There's blue light here, dark, and we can also use green, we can use red, we can use near IR. And the idea is to have an entire bioreactor containing, let's say, in this example, 4 different strains of bacteria and yeast, one growing under red, one growing under green, one grows under blue, one grows under dark. So you can actually, just by having different light cycles of green, red, blue, dark, varying light cycles, you can actually modulate the population ratio of each of these microbes for maximal chemical production. Now, I'm not going to delve too much into this, but we do have a way to control baker's yeast with red light. So this is just harnessing a split transcription factor fused to a red light switchable red light optogenetic tool. So we're actually able to cause red light inducible gene expression with some of the tools we have in hand. This one here is a blue light inducible optogenetic tool that previous PhD graduate has utilized. So in theory, we can combine this blue light tool and the red light tool and have several different members in a microbial co-culture, control with red, control with blue, and control with dark. But what I want to focus on is bacteria. Now, bacteria is one of the most, or the most understood and well-studied microorganisms out there. Bacteria has been used to make drop-in biofuels, precursors to a polymer, as well as petrochemical. And E. coli can also be used in microbial co-culture, like I mentioned before. So you have a bioreactor containing several different strains of E. coli, each making a certain intermediate to afford one final product. Now, bacteria grows fast compared to yeast. And we need really a good way to suppress E. coli growth when we want, and up E. coli growth when we also want at another time. So this is a previous work done by Dr. Makoto Lawani, where if I just go through this carefully, in the dark, this YF1 protein acts as a protein that can phosphorylate FixJ. Now FixJ, when it's phosphorylated, binds the FixK2 promoter, which makes lambda repressor, as well as MOSF, which is a toxin to bacteria. So when you, in the dark, a lot of toxin is made, so E. coli don't grow, right? And when you shine light on the right side here, that same protein is a light-sensitive protein that can now act as a way to remove phosphate group from fixed J. Now, when it's removed, it doesn't make lambda repressor nor the toxin. Instead, this antitoxin is made, so allowing cells to grow. So you can have various ways having blue, dark in controlling really the growth rate of E. coli very easily. So what I've done is hijack the system and use red light instead. So we have a same set of tools. Here, this is a photoswitchable protein called DRB-PHP that changes conformation with red and comes back to its original conformation in near IR or dark. This, when it's in near IR, when you shine near IR, this protein can phosphorylate fixed J again, binds the cognitive promoter and makes toxin. When you shine red, this now removes the phosphate group again, and the same idea no longer makes the toxin or lambda repressor, now allowing antitoxin to allow cells to grow. So what we've shown here on the left is that red tool that I've explained to you in the previous slide. In red light, this thing grows just well as just having empty vector. Now, when you have blue light, this also responds, which is unfortunate, but that can easily be inhibited just by shining near IR concurrently. And in the dark and just near IR, almost no growth of bacteria. Now, OptoTA is that blue light system that I've mentioned to you before that we can use in conjunction with red on TA in a microbial co-culture. Only grows with blue and doesn't really respond to any other light besides, unless you have blue light, it doesn't grow at all. So we can then, and with this red on system, I also show that this is tunable. So tunable expression is important because if you're just going to shine light and it's going to grow like crazy without any sort of control over how fast it grows, it's not going to be useful in microbial co-culture. So having lower percentage of light input into the system, you can actually lower the, in this case, it's GFP expression, which would correlate to growth if we start using the toxin-antitoxin system. We show that this is also tunable with more amount of light input into the system. So with this, we're at a position where, back again to this diagram of bioreactor containing different strains, we have a way to control one bacterial strain's growth and inhibition of growth with blue light and dark. Another one, control that with red light and inhibit the growth with near IR or dark. So that's where we're at with that. So now the idea is to be able to put in a fragmented biosynthetic pathway, put one part of the biosynthetic pathway into one of these, put the second part into another, and really control the growth and population ratio dynamically. Because with light, you can actually shine whenever you want. You can turn it off whenever you want. So that's the idea with what we want to do with this now. And for the second part, I want to go back to yeast. Now, I mentioned Baker's yeast a lot because it's a model organism for yeast research. Isachenkia orientalis is also is a non-conventional yeast that has been getting a lot of attention recently. Isachenkia orientalis compared to Baker's yeast or Cerevisiae is attractive because it has high tolerance to temperature as well as low pH. So you can actually use these to do metabolic engineering at lower pH, and it would be advantageous to make things that are chemicals that are acidic in nature. It's also advantageous in low pH fermentations or a process called SSF, which is basically using enzymatic hydrolysis to break down cellulosic biomass, and at the same time use yeast to eat up those broken down sugars to make to ferment to produce chemical of interest. Now, because of the fact that Cerevisiae can't really handle low pH, when you use Cerevisiae for metabolic engineering, you do require neutralization process, which is which can be harmful to the environment. With Isachenkia, this neutralization process can be bypassed. So what we want to do is use Isachenkia, as you see in this diagram below, to make this molecule called citramalate. Now, what's citramalate? It's a precursor to PMMA, polymethyl methacrylate, which is used to make acrylic glass. PMMA is actually expected to reach about 8 billion in market size by 2025. And people have tried to produce citramalic acid or citramalate in E. coli. But then the problem is with E. coli, you also have the same problem you see with baker's yeast. You have to neutralize after you've made enough citramalate, which actually increases the environmental footprint by 30%. So what optogenetics got to do with this? Well, we can use optogenetics not only to control population ratios of microbes, but we can use it to increase gene expression of the enzymes within the pathway of citramalic acid. So the idea is, I put a dot here for our collaborators' reasons, but you can use enzymes that actually use pyruvate and acetyl-CoA to make you can light control enzymes that use pyruvate and acetyl-CoA to make as much citramalate as possible. Basically, you can upregulate this enzyme or downregulate this enzyme at whichever point you'd like with using light. But before we do that, well, how do we know this is even possible? So what I've done up till now is to use a existing blue light switchable gene expression tool I've shown you at the beginning of the presentation. So it's called OptoEXP, where it has this blue light inducible transcription factor, EL222. Now in the dark, the DNA binding portion is docked against the protein. So it actually doesn't bind the promoter, doesn't make anything, any gene of interest that you have downstream. Only when you shine light, the protein unfolds and dimerizes, allowing this DNA binding domain portion to bind the cognate promoter, making gene of interest. So far, what I've done is put a GFP in just to see if this thing works in Isachenkia orientalis. And remarkably, this OptoEXP thing right here, in the dark, you actually don't see much expression of GFP, and with increasing doses of blue light, and this percentage, I mean that maybe it's kind of hard to see, but the down here is 1%, meaning 1 second on and 99 seconds dark, and 10% meaning 10 seconds on and 90 seconds off, and 100% meaning full blue light throughout the entire process. So of course, you expect that higher the percentage, the more blue light dosage you get, and this is again tunable. Now that's important again, because if you want to control enzyme expression within the pathway to really optimize citramalate synthesis, you need a way of really inducing it at varying levels. And just as a comparison, we have Isachenkia, a very strong promoter in Isachenkia orientalis as a control here. Now compared to that, our expression is much higher. So it's a positive sign that we can actually use this tool in really using this to do light control of enzyme expression, levels of enzyme expression in Isachenkia orientalis, really for not only for citramalate, but various other organic acids that are very difficult to make using E. coli or simple baker's yeast. So that ends my presentation, and I thank my lab for being here.
We have a little less than 10 minutes for questions. I can start.
Yeah.
So what chromophores do you put on the proteins?
Yeah, so this blue light tools, the reason there's actually a lot of blue light tools in literature, and the reason is it uses FMN, flavin mononucleotide, which is everywhere across the animal kingdom. The problem with the red light tools is it uses something called phycocyanobillin, PCB, which is available in plants and cyanobacteria. So those actually have to be exogenously added to the culture. There are lots of ways to actually co-express the system, co-express the chromophore synthesizing machinery at the same time. So there's been efforts in that. Yeah.
So are they covalently linked to the protein?
They're covalently to the protein. For red light system, it's always covalently linked. And the way it photo switches is you shine light, it goes through ENZ isomerization within the chromophore, and that's enough to cause the conformational change. The one with the blue light tools that utilizes FMN, these actually are, from my understanding, in dark, it's not covalently linked. And when you shine light, it links covalently. So that's reversible.
But it's in a complex. It's just not covalently.
Yeah, it's a complex.
It's not floating free in solution.
Yeah. It's always kind of like, it's bound pretty well to the protein. Yeah.
Thanks.
Yeah.
I know that you said like Isochankia, like two of the benefits are it's more like tolerant to high temperatures and low pHs. Like what actually are like the temperatures and pHs that it is tolerant to compared to Cerevisiae?
Yeah, so Isochankia, from my understanding, what the last time I saw was it was able to tolerate pH 2.
Okay.
Temperature, from my understanding, was 42.
Do you know how the pH setting work with Cerevisiae? Like how does that compare to what Cerevisiae uses?
I think you would have to go, I think even pH 5, I think is Cerevisiae. Yeah.
I had another question.
Yeah.
Anybody else does? You have at least on one side somewhere. Yeah, it's like different percentages of the different
Yeah.
wavelengths of light. Instead of just the percent as a fraction at the time being irradiated.
Like here, or
Well, maybe it was the different fractions of the time. If you ever have two lights on at once is my question essentially.
two lights on at once. Yeah.
Then change the fraction of each one.
Yeah, that's actually one of the beauties of this is that you can do that with let's see. This what I have here is 20 seconds of red and 20 seconds of near IR followed by it in cycles. Now, but in theory, what you can have is change the intensity as well, but have two lights on at the same time, and you can use that to tune as well. I'm not sure if that's your question.
Yeah, that's my question essentially is instead of doing it on a time-based percentage, percentage of the intensity of each light can be used to tune them too.
Yeah, intensities. Certainly. It's basically how much photon gets in there most of the time. So intensity, if you lower the intensity, that should also, it's something that's hard to predict though. So it's something that we're always going to have to test, like what intensity to use.
Yeah, and a related question to that is essentially, this one is kind of convincing that it's essentially changing the intensity of the light, but you're mixing with a different light. But can you modulate just with a single wavelength of light? If you change the intensity and just keep it constant, is there a threshold? Essentially, is there a threshold where if it's just on, you just start getting full growth, or can you get
You mean like
fractions of growth by?
Yeah, you could
turning
do
the
also
light
do
there.
that. Yeah, it's something hard to predict, but like it's possible. Like I haven't tested with this one yet. It's really dependent on your system, like your light sensitive protein.
Okay.
How sensitive is it to certain?
Yeah, it may just.
And it also depends on the wavelength. It's possible.
I think it's kind of goes in the line of what you're saying that it's hard to predict, right? To see exactly what's going to happen. Are the kinetics of, I guess, if you wanted to build a model of the bacterial populations and put them in a system of differential equations or like that, are the kinetics well characterized? Has that been studied? Is there like a big void there that just unknown, or
kinetics of like how they're growing?
Yeah, like what are the rates of growth?
Yeah, that's
how is light affecting the rates of growth?
Right, that would be something we're going to do. You know, there's work being done in our lab doing that as well, and several other labs out there. So now we have the tool, which, you know, seems to be growing and not growing with light, and that's certainly our next step. Yeah.
If there are no more questions, thank Jaewan again. Our second speaker today is Dr. Yaguang. Yaguang earned his BS from Nanjing University in Environmental Chemistry and his PhD from the Department of Energy, Environmental and Chemical Engineering at Washington University in St. Louis. He joined Princeton in 2022 as a postdoctoral research associate at the Andlinger Center, working with Professor Kelsey Hatzman. While Yaguang's doctoral research focused on nanoscale interfacial control of ions and calcium carbonate formation in environmental systems, here at Princeton, he's exploring engineered approaches to carbon capture using novel materials and processes that aim to lower the required energy for this process. Today, he'll be presenting on this in his talk, Understanding the Critical Role of Anions in Moisture Swing CO2 Capture. Please join me in welcoming Dr. Zhu.
Great, thanks for the introduction. So hello everyone, I'm Yaguang, and I'm a postdoc in Professor Hazel's lab. So I'm joined last year, so we're starting a lot of new tools trying to do better separation, including carbon capture from air. So today I'm going to show you about moisture doing carbon capture. I want to, everyone after this talk can understand why we care about carbon capture and why we need to use the moisture to do the application and how we can conduct the experiment to test whether those materials can do the moisture swing or not. And finally, I want to show you where we can improve the performance by anion engineering. So now let's start. I think everyone here has heard about climate change and we have too much carbon dioxide emission. The major problem of the too much carbon dioxide in the air is we have an increasing temperature from the pre-industrial time. And then everyone learns about 1.5 degrees Celsius. Why is that 1.5 degrees Celsius important? It's because if we don't do any interference, like we have in more and more carbon emission by the year of 2100, we will have about more than 2 degrees Celsius increase. So if we can have some new strategy to reduce carbon dioxide or capture carbon dioxide, we can maintain an increase only to 1.5 degrees Celsius. So that number determine how much effort we have from today to like 2100 to reduce carbon dioxide. Currently, we still have a lot of carbon dioxide emission every day and then every year, and it keep increasing. The problem is we still need a development. In order to achieve 1.5 degree by the year of 2100, we need to achieve near zero emission by the year of 2100. We still generate carbon dioxide. What we can do to have a near zero emission means we need to capture them. So this gives you the critical point that we need to capture carbon dioxide, find a new way to capture them, or at least new carbon dioxide to capture. So where the new carbon dioxide can come from is air. So I'll introduce you the technique called direct air capture. Most of carbon capture in current technology is captured from the power plant. They have a lot of carbon dioxide emission there, so it's easier to capture. But by the year of 2010, some people think that actually we probably can just capture carbon dioxide from the air we're breathing because there still have 400 ppm there. More importantly is most of the carbon dioxide emission goes to the air and then they keep increasing. So before we do any experiment or research, let's think about whether that's feasible or not, we capture them. So there is a thermodynamic calculation that how much energy we need to capture carbon dioxide directly from the ultraviolet air. So here is just an equation showing the minimum work that we need to use to capture them. The comparison is capture carbon dioxide from the power plant that need 10 kilojoule per mole of carbon dioxide capture. If we just move it to the current technique to the air, theoretically, we only do double this energy. This sounds great. It seems workable. So since then, people start to moving all of the current carbon capture technology to the direct air capture. And then the problem comes. The first thing is if people just use the porous material such as active carbon or metal organic framework, those porous things to capture carbon dioxide from air, they are very low concentration there. So the water vapor, the nitrogen, or other gases all compete for the surface area there. So the efficiency is super low. And then people notice that, yeah, we probably need to find some better material, have a stronger chemical binding with the carbon dioxide. So people move to the chemical absorption. That there are a bunch of chemical absorbents, such as ionic liquid amino acid base, or some electrochemical way. The key thing there is people think we need a strong binding between the materials and the carbon dioxide. At this moment, you may think that will be good. We need this high selectivity of those chemicals for carbon dioxide. But the problem is this can be good and it cannot be good because in addition to capture the carbon dioxide from air, we also need to regenerate the material. We need to release them again. This entire process is not only capture. The entire process is capture first, release later, and then to have a regeneration of the material. But because we have a very strong binding of those materials with carbon dioxide, that's typically the chemical bond. So we need a huge energy to regenerate the material. According to a report by National Academy of Science, Engineering, and Medicine in 2019, just by using the current technologies to regenerate those materials can cost 8.4 to 12.5 or 4 to 6 gigajoule per ton of carbon dioxide capture. Those high energy costs make those current techniques are not attractive because we want to reduce the carbon dioxide by a lot of money. No one will pursue that. We want to find some way that we can capture carbon dioxide selectivity, meanwhile regenerate it with less energy expensive. So here comes to the new technique show up called moisture swimming carbon capture. This would be accidentally funded in 2010 because people initially don't want to test with the moisture swing, but later they found actually it works. The major principle is this technique regenerates the absorbent only applying moisture or just applying water there. So according to calculation, they have a least energy cost when they try to do the regeneration process. Such as plot shows here, if people using amine or guanidine or some strong base to capture carbon dioxide and release, they have an energy cost a lot when doing regeneration. The only small one is the physical absorption, but as I mentioned, physical absorption is not ideal for direct air capture because the CO2 amount there is so low. For this moisture swing carbon capture, people notice that they can use a material. I will show you later what the material is, but they can capture carbon dioxide when it dries. But if you increase in the relative humidity to like 70 to 80%, those absorbed carbon dioxide will start to release. So now I'm going to talk about more about what material can be used for moisture swing carbon capture. That's typically the ion exchange resin. Previously, people used this type of resin just for cleaning water because they can exchange ions inside the water. But later on, people started testing with carbon dioxide on this new thing. The resin, if we just purchase from any vendor, we'll just like a sphere. And then if we zoom out more, it have a lot of porous structure, enable the water or ions or gas such as carbon dioxide to diffuse. And then even we do more, we'll start to see the polymer chain. And then this polymer chain are composed of the quaternary amine to provide the positive charge. And then we use the anion exchange resin. So anions are freely to exchange there. For this moisture swing carbon capture, people start to notice that is probably we can use carbonate to achieve this goal. So, and then everyone just think, oh, that's super good. Let's just try it with carbonate and let's just try to different way to have a better performance. So what they did is they think, how about we change the polymer, we change how we design it, how much pore we have. So indeed, it works. So the earliest works start from 2013, and then people find new way to synthesize the polymer with super high surface area. They have a lot of branches there, and then they have a lot of surface functional group, and then that indeed works. And then people also find that if they change the pore size of this resin, if they have a pore size about 10 nanometer, it works for the moisture swing carbon capture. But if they have a pore size about 100 nanometer, it doesn't work. Also, if people try and say we adding more functionalities, hydrophobic or hydrophilic functionalities into the structure design, they can change the stability of this resin inside different moisture level to see whether the improve or decrease the CO2 amount. But in 2019, a question comes up, why those materials can work for moisture swing carbon capture? Because we know that it's selective for carbon capture means there's a chemical bonding there. But why this chemical bonding is so easy to break if we just applying water? And then there under it, resin indeed is important, it provides surface area. But the anion there actually matters more about this mechanism, why moisture itself can changing the carbon capture or release. In their modeling result, they compare two scenarios. One is just using sodium carbonate, another one is ion resin with carbonate. The first conclusion they have is they indeed need to have a pore, but that's only the ion exchange resin provide, it's just a pore. The more important thing is the chemical reaction shown here, the carbonate hydrolysis reaction to form the bicarbonate and hydroxide. This reaction, if they not happen in the pore, it's not reversible. But in the pore, it is reversible because of the water amount. In their modeling, you can see if the water amount is a little, just like or 1 or 2 water molecules there, the overall reaction energy of this equation will be negative, means more hydroxide will form. That means more carbon dioxide will be captured under low moisture level. However, if they have more water contained into the system, for example, let us show about 10 water molecules there, the reaction energy will become positive, means the reverse reaction will be preferential. In that way, hydroxide will decrease, carbon dioxide will start to release. So by knowing that anions matters more, our research question is how different anions will affect this system and how they are interact with water to make the process better or worse. So now I'm going to show you how we do those experiments with our setup to test whether or not they can do the moisture swing or whether or not improve the carbon capture behavior. So I have a bunch of stuff here to simplify. I will introduce you one by one. So clearly we need to have an analyzer to tell how much carbon dioxide and water there. So that's called CO2 and water analyzer. So here just a video showing if we can changing the moisture inside the carbon dioxide will correspondingly change slowly. So in the time of about like tens of minutes or even hours. And then in our study, we need to control the humidity because as you know, it need to have a different humidity to capture or release carbon dioxide. So we have an instrument called dew point generator to set the initial humidity first. It can either to reach a very high humidity. This number means a dew point. If it's equal to the room temperature, means 100%. Or we can decrease to the low humidity. As we know, we want to capture carbon dioxide first. So let's move to the low humidity and make the air have a low humidity, like about 32% humidity, and flow through the resin we have to see what we can get. So with the time evolution, carbon dioxide is near zero because they are absorbed by this resin and started to go back to the air concentration of 415 ppm. Now we are confirmed at low moisture, carbon dioxide is indeed captured. And the next question is, can we have this reaction reversible when we have the high moisture? In that way, we'll introduce you another thing, how we can control the moisture quickly is using the Peltier cooler. So in this instrument, I will show you a short video to show you this instrument can quickly freeze everything surrounded within a few seconds. So here you show you just the ice freeze outside there, but the the gas flow inside will also have the water condensed there. So they can quickly go to the zero humidity. So now I'm going to show you, we can change the humidity from high to low and then back to high, and to see how the carbon dioxide change in the system. So we can see when the humidity is high to low, we start to have a carbon dioxide decrease, means carbon dioxide is captured. If we back to the high humidity, this carbon dioxide will start to release again to back to the same level. And then we do multiple cycles, it clearly works. So now we have the baseline. We know this resin works, carbonate resin works. What about we have other resin? So our strategy is very straightforward. We have the resin and exchange with different kind of anions that we know. For example, we can change to phosphate, sulfide, borate, a lot of things. And then in our study, we choose the 5 of them. Carbon I already show you. We have phosphate, sulfide, acid, and borate. And then we choose those because they have different basicity. Anions have basicity because they have hydraulic reaction. We expect phosphate behave better than sulfide, than carbonate, and then borate. So, but is that really the case? So first we see how much carbon capture initially at the dry condition. So this is within our expectation because phosphate is higher than sulfide and higher than carbonate because they have higher basicity. And however, if we're trying to do the moisture swing, the result is not all the same. Because for carbonate and phosphate, we indeed see when we change in the high humidity and low humidity, the local concentration change a lot. So you can see most of the captured carbon dioxide at dry condition can be released under wet condition to do this moisture swing capacity. However, only for sulfide, it captures carbon dioxide, but it cannot release. What happens there? Let's back to the reaction. For this reaction, in order to have the moisture swing behavior, it captures carbon dioxide because it has a hydrolysis reaction to generate hydroxide. But it needs to release carbon dioxide when under high moisture level, the reaction needs to go backward. For phosphate, modern people suggest that indeed they can have moisture swing behavior because both phosphate or monohydrogen phosphate will have this type of reaction. Under low moisture, reaction energy is negative. Under high moisture, reaction energy is positive. However, for sulfide, the calculation shows that a free energy of this reaction will always be negative, no matter how much water you add into the system. So that means the ability of this hydroxide to decrease at high moisture determines how much of the released CO2 can be released. And then we'll move to another anion. This time it's borate. Initially, when we check the capture amount is low, the capacity is low. So we thought it's worse. But if we just check their kinetics, we found that just adding borate itself can reduce the kinetics by 20 to 40%. Even more, if we adding a small amount, like 20% of borate into the carbonate, they can also decrease the reaction, the kinetics a lot. It's by no means we add a worse thing into a system can make the system better. There must be some reason for borate that they place a different role there. So that's sort of interesting what role they place there. So still, with two extremes, we have carbonate and borate. We know borate works, but why we adding a little bit borate, they can make the behavior looks better. We look from some literature actually in other systems for capture carbon dioxide. People also notice that adding borate can enhance the kinetics slightly. This may indicate that borate have a different reaction there. And then this time the reaction is shown there that borate serves like a self-catalyst. This time the borate can just interact with carbon dioxide and water directly to transform to bicarbonate. So in that way, we should see when we add borate to the system, the water will have a strong binding with borate. So indeed, what we try to test is if we dehydrate our resin to see how much water it can retain. And then the blue bar shows you how low the water can be extracted when we go to low humidity. If this is very high, means the affinity of the water to the anions is low. If it is pretty low, means the affinity of the water to the anion is high. So we can clearly see that only borate have the lowest strong kind of dehydration rate, indicating this mechanism works. This means borate takes another role to enhance the carbon capture, they try to react with carbon dioxide and water directly to capture more carbon dioxide. So with that, a little bit conclusion is, in our study, we evaluated how anions as a major role in the moisture screen carbon capture can change the entire capacity and kinetics. And then we found that increasing the basicity is not the only way. We need to make sure those captured carbon dioxide can be released. And then borate itself takes another role, and then they can also enhance the carbon capture. So currently, we're also trying to improve the system more. So such that we are trying to capture water together in this system. And also we are trying to have some new way to trigger this water uptake and water release. So thank you all for listening. I would like to take that.
Good questions?
So what sort of energy reduction do you expect from in the release process by using borate or the moisture sinking process?
So currently, let's take one example. If we're using sodium hydroxide to or calcium hydroxide to capture carbon dioxide, we will form a new solid such as calcium carbonate or like sodium carbonate. So if we want to regenerate this material, like back to the hydroxide, the only way is we increase in temperature. That's typically reaching about 300-400 Celsius. And then according to calculation, that will cost about gigajoule of the energy per ton of the carbon dioxide capture. By using water, like in our system, what we do is we have a water source, it's just increasing the relative humidity locally and then increase that. So in the real application, potentially you can just pour water on it and then we'll start to release carbon dioxide. So that's what we define the reduction of energy that's caused to regenerate those orbits.
So you would not need any combustion, basically.
Correct. Correct. And then combustion is not the only way to regenerate. Like some other people try to use the pressure, right? Like they use a very low vacuum and then they can, but still that costs a lot of energy.
What material is the resin made out of?
It's basically just the carbon polymer. So any carbon chain with or without bending ring or polymer can work, but they need to have a positive charge there because we use anion as a major role. So that's why we show they have a quaternary amine to provide a positive charge of the backbone of the polymer. And then you provide the anion as a major role to play the moisture thing.
And how is the materials, the resin material synthesized? Is this whole process like net zero?
That's a good question. So based on my information, those resin are pretty cheap because we can get 1 ton about $100. And then those resin typically are used for water desalination or water cleanness kind of thing. So considering the entire project, it's a good thing that I should check how much energy it costs to generate this material.
And are they biodegradable?
I don't think so.
Yeah,
Because there's a pull of carbon chain and the bending ring, so I don't think it's biodegradable at the moment. But I don't think that's disposable because for those ion exchange resin, you can just use it forever. We just need to use the anion there.
yeah, I was gonna say that. I was gonna say in water treatment applications, they get fouled over time. But for this, I think they could just keep going and going.
Yeah. And then as a regeneration, it means we just apply water. But if we really want to clean this, we can just put in seawater to it. Because seawater have a lot of chloride, they can clean everything inside. And then we can just replace it with the anions we want. There are indeed some people trying to see if they can regenerate the material just by seawater recently.
I was wondering, in trying to maintain waste at a high humidity, or high moisture, low moisture system, any like actual energy?
Yeah, so in our, that's a good question. In our experiment, the answer is yes, because the facility will have a humidity inside our building, but we need to change the humidity through our instrument. For example, we have an instrument called dew point generator. They can change the humidity locally. However, in the reality, if we really want to apply this, we don't need that because typically in all the regions across US or entire world, the daytime, I mean in the morning or in the nighttime, the humidity is different. So that is large humidity difference enough to do this moisture sweep. So that is the initial, actually there is some startups start working on this, just apply to at Arizona or someplace. Their temperature of the daytime and nighttime is a larger difference, so they can already change it enough humidity there.
And what is like the reusability of this kind of material? Can it be reused like many times?
Yeah, I mean, we didn't test to reuse that, but based on my information or based on my knowledge, this material theoretically can easily be reused without any degradation because the material itself is made from carbon chain. That's pretty strong. And then the only thing is anion. And then if get some contamination, for them, some anion doesn't work for moisture swing happen, we can just replace or exchange those anions with the anion we need.
So another question was about, so you're saying you can potentially not have to supply any humidity yourself, you can just rely on swings in the ambient temperature. But how much does that slow down your cycle time? Because if you're using heat, you're paying for power or energy, but you can more quickly regenerate and keep the process moving. Whereas if you're waiting, so is that a big trade-off or?
I agree. I think the kinetic part matters a lot. But currently, so first thing for the, if we're doing the combustion or other things, the typically time scale for regeneration is still about hours. Because even though the reaction happens fast, like let's say we degrade it at about 700 Celsius, the degradation probably takes about 10 minutes. But increasing to 700 Celsius, you need a few hours. And then back to the normal temperature, that's a few hours. And then for this release process, what we test is typically also a few hours, like 1 or 2 hours. They can, under high moisture, will start to release. So just about the time-wise, I would say they're close. And then still the major concern right now is not the kinetics, because we have a lot of air anywhere. We don't worry, sometimes don't have air. But the only thing is how much we can capture and how much energy and cost we use for capture those.
So for the borate, it sounded like you're saying that the primary limitation in that is actually it's slowing down the dehydration rate.
That's a kind of a side proof. So the basic idea is we think borate itself cannot be a good anion to do the moisture swing capture.
Right.
But when borate adding into carbonate is good. So they can enhance the performance of carbonate. Means borate have another mechanism to enhance the carbon capture. So just checking some literature, we found that it's highly possible that borate itself serve as a catalyst to directly make the carbon dioxide and water react. So in that way, when borate is in the system, we expect the water is hard to remove. And then we use the experiment to show that indeed, when we have borate in the system, the water is hard to remove. So indeed, that mechanism should, or at least may work for our system.
So yeah, so that makes sense. But theoretically, then you could dehydrate by increasing the temperature a little bit, and you might have a higher capacity with borate.
I actually that yes, but increasing temperature costs energy.
Yeah.
So that's why one of our alloy we are trying to use some solar energy to increase temperature. That's also one of the future work we are like working on.
Yeah. These resins are white colored, right?
Yeah, so that's why we moved to another resin, which is not resin, another particle which is black.
Sorry, one more question. I also work on that. So what are the costs of these materials compared to like more conventional, like potassium hydroxide or other like liquid?
Yeah, I mean this expansion happened. Actually, I don't have a very direct value of those price now, so I wouldn't say this much cheaper than the sodium hydroxide or potassium hydroxide. But just for my information, this resin also pretty cheap. So if you call to the dollar company, and then if you want to add 1 ton, the potential can give you tomorrow that if you just spend about $1,000 or $100, because as I say, those resins are not for carbon capture initially. Dow inventing them for the desalination, and then those are already a lot of companies use that for desalination. So I don't think production can be a problem. Also, there are a lot of like storage of them anywhere. We just use that for a different application. But that's a good point. We can start to check what is the cost compared to the sodium, like the base, how much cost they have.
And like at a large scale, where do you anticipate the major costs to be for this type of system?
I think two major costs for the moisture swing that we have. Firstly, is the production of the resin itself. The second one is we still need to dry the resin. Currently, our way to dry the resin is we apply the dew generator or pellet cooler. Then in reality, I think people still just flowing dry air through it. That still costs energy. So one of our future work is still say if we can use solar energy to increase the local temperature to dry the particle. That will be something that we're working on to further decrease the energy and then the cost.
Can I ask, I might have missed it, but I was gonna ask about the sequestration side. So are you releasing carbon into air and then pressurizing that and injecting it, or do you release it into water that you then have to dispose of?
Oh, okay. So for this moisture swing, actually, it's not really in the water, it's still in the air. So basically what we show is we have the swing. You remember at high moisture, it goes to about 300 ppm. But that we just add about 0.2 gram of the resin. Let's say we have 200 grams. Then potentially we have a million or even 1,000 or probably even million ppm. And then that's with a high concentration of carbon dioxide. And then we can just capture them in the tank.
Okay, and then you.
And then there will be already Python purity of carbon dioxide, and then treating that would be much easier compared to ultrasonic.
So it's like all the reaction.
Sorry, all the reaction will happen in
Is
one.
that that reaction happens? Are they like happening?
In our system, we have just a small beaker to make the reaction happen. So if we're talking asking about scale up problem,
Yeah.
I think when we have scale up problem, just to make because first thing, those resins are highly porous, and then those are small. So I don't think water cannot permeate them, or gas cannot permeate them. So as long as they are just in a jar, we can make sure the water vapor can penetrate or go to the deeper part of the jar. That reaction should work. But at least our work here is we are testing in a small system.
Yeah, I would imagine it's a packed bed two packed bed.
Yeah, our pack bed is very tiny. we are not testing a very huge pack bed. Because it just tries to show anion make difference. So yeah.
All right, thank you.